Alright, so in this review video, we're going to be talking about mutations and genotype and phenotype. So just to first start, we'll talk about what mutations are and how they occur. Mutations are known as any heritable change in genetic material. This means that it can be passed down via cell division. Mutations also span a range. This means that the mutation can just be a simple, just a nucleotide change, like a substitution, insertion, deletion, or something more like the deletion of segments of chromosomes, which are on a larger scale. So the way mutations occur is they occur by a mistake that's made when DNA is replicating, and when that mistake is made, then successive replications will have that mutation, and will no longer have that normal DNA. And also, a lot of environmental factors increase the chance of getting a mutation, such as chemicals or radiation, like X-ray and UV rays. And once you get into OChem, once you start doing the OChem lab, there's a lot of chemicals that are classified as carcinogens. This means that they have the ability to cause cancer by mutating your DNA. So another thing is about mutations is they occur on the basis of chance, meaning that they are random. So a quote that was from the Cogbooks reading says that the role of the environment is not to create specific mutations, but instead to select for them, which is very important to know. And lastly, mutations are fairly common in humans compared to other organisms like bacteria. The reason for this is because we undergo a lot more cell replication and have more genomes, so that increases the likelihood of a mutation. That increases the likelihood that a mutation will arise. Even though mutations are more common in humans, it does not affect us that much because only 2.5% of our genome will code for a protein, and the rest are we don't know a lot of it, but they don't seem to do any harm. So that is mutations and how they occur, and then we'll go on to next talking about small-scale mutations. Alright, so small-scale mutations. First we'll talk about substitutions, and then we'll go on to talk about insertion and deletion and kind of frame shift mutations. So if you recall, central dogma states that information travels from DNA to RNA to a protein. And the specific bases that are found in the RNA are going to come from the coding strand of the DNA. And if there are any changes made to that coding strand, or actually a template strand is what is transcribed, but the coding strand is the same as a transcribed template strand. But anyway, if something is changed in the DNA, like base pairs are changed, it's going to alter our final protein structure. And so for substitutions, there are three main types, and we'll go through them. So first, we're going to talk about our synonymous mutations. So actually, before we talk about that, we're gonna talk about non-mutant. So like what this is gonna look without any mutations. So we have our DNA right here. It is transcribed into this mRNA, and that mRNA will code for this specific polypeptide chain and this sequence of amino acids. Right. So here we have a proline, glutamine, I think. So now, if we look at our synonymous mutations, this highlighted part right here in red, so this A and T is changed from the non-mutant, which was a G and a C, I believe. Yeah, G and a C. So when that change is made, we're gonna have a different base in our RNA for our second codon, we can say. So let's say this is our first codon, and this is our second codon right here, the GAG. So for a synonymous mutation, our second codon is going to be GAA. But this type of substitution, this type of mutation, does not cause a change in our amino acid sequence. So why is that? Well, if you remember our codon chart, it is important to know that that codon chart is very redundant, meaning that different codons, different combinations of a codon will code for the same amino acid. So in this case, our GAG and our GAA, if we were to look that up on a codon chart, would both code for glutamine, right? And that gives you an overall same amino acid sequence. So that doesn't have any effect. So synonymous mutations has no effect. It is often known as a silent mutation, right? Because you don't notice a change. It's silent. So moving on to a non-synonymous mutation. Let's see here. So in a non-synonymous mutation, we actually do have a change in our amino acid sequence for our polypeptide. So as you can see here on the second codon, we're going to have GUG, right? The change is made in the DNA, and that's going to result in a uracil here instead of an adenine. And the GUG, unlike the GAG in the non-mutant, will code for a completely different amino acid. In this case, valine, I believe. So what that does is it alters that polypeptide chain, and that results in a misfolding protein or a different way of folding that protein, and you're not going to get the same outcome. And this type of mutation is called a missense mutation. Okay, so those are two so far. And then lastly, we have a mutation known as a nonsense mutation. So what a nonsense mutation does is we have different base pair in our DNA now. So instead of a G and a C right here, we're going to have a T and an A, right? And when the mRNA is transcribed, we're going to have a U here on our second codon to begin with instead of a G. And what that does, it results in a stop codon. If you remember, again, that codon chart, there are there are 3 stop codons. And what a stop codon does, it just stops the synthesis of the protein altogether. So it will start from proline, and then once it hits that stop codon right away, then it will stop. There's going to be no more protein. And nonsense mutations are deadly because of that, right? Because we're not going to get any other amino acid chain. So usually we would have a GLU and a GLU right here, but now we don't have anything else, and we just have our stop codon. So frameshift mutations can be either insertions or deletions. So let's look at like an analogy for this real quick. If we are, see if this works. Okay, so over here. So let's take a look at this sentence. So the big boy saw the cat eat the big bug. All right, that's our that's going to be our mRNA. All right, so just kind of analogy for it. And notice how they're going to be grouped in threes, just like how the mRNA will be read in known as codons, right? And this is like the reading frame. So we're going to read THE, BIG, BOY. So it makes sense, like how we read it, right? For a normal sentence, the big boy saw the cat eat the bug. However, if we take out the E right here, right, that results in a frame shift, meaning that we're still going to have to have these letters be grouped into threes. So, but because of that deletion, the mRNA or the sentence won't make sense, right? So you can't really read this. It doesn't say anything close to what our original sentence says, and that's because it is a deletion of a nucleotide or a letter that is not in units of threes. And this can be for insertions as well. So what I kind of am getting at is, like, let's say that instead of just deleting one letter right here, instead of just deleting the E, we delete the entire word the, right? So the TH and E. Our resulting sentence would just be, big boy saw the cat eat the bug. That makes more sense than whatever this is that we got when we just deleted the E. So hopefully that makes sense. But okay, we will talk about now, first of all, a deletion, an example of a deletion. So this example relates to cystic fibrosis, a disease known as cystic fibrosis. And basically there is a protein known as a CFTR protein that is essential for a normal person who does not have cystic fibrosis. So it's responsible for transporting chloride ions, right? So, and our DNA that is responsible for this, it is transcribed into the mRNA right here and translated into our protein. Okay. So for cystic fibrosis, we have a mutation in the CFTR protein. So in our DNA, actually, we're gonna be missing this three codons right here, or three nucleotides, and that's just gonna only result when we transcribe our RNA, and when we translate it into a protein, that's only gonna result in our just our 2 amino acids instead of 3. And when this happens, basically our protein that's responsible for the chloride ions, or the CFTR transporter, is no longer there. So there's nothing there, right? Because it is a inactive protein, and it just kind of degenerates because it's not the full thing. And that is how deadly a deletion can be. So that's one example of a deletion. So next we're going to talk about an insertion. So right here we have our non-mutant DNA, okay, and it is transcribed into our mRNA and then our protein, and this is our amino acid sequence that we get. All right, so now we're going to have a mutation. So we're going to actually insert the CG base pair right here. So if you look at this non-mutant one here, it's going to go CCT and then GAG. But since we're inserting that, this one is going to read CCT, and then we're going to have that extra one, and then it's going to go CGA, right? So, and so for our mutant strain, this is going to be our transcribed strand, also known as a template strand. So our mRNA is going to transcribe that, and we're going to end up getting this for our mRNA. But since we have that insertion, our reading frame is going to shift, like I mentioned before, because it's adding in, it's not adding, let's say, in pairs of 3. So we're going to have, after our first amino acid, everything else coming after it is going to be different than what we had for our non-mutant strand. Okay, so lastly, I want to talk a little bit about genotype and phenotype. So a genotype is just our genetic makeup of a cell, like chromosome, DNA, right? And a phenotype is an individual's observable characteristics. And something important to note that is phenotype is also influenced by the environment. So we can say that a phenotype is a gene environment interaction. So an example of this can be like skin color. So let's say you have a pale skin, and like that's your genotype, but you are out in the sun a lot, and the environmental factor of the sun influences how you look and how your skin complexion is. So you're gonna have more of a darker complexion if you're out in the sun a lot. Yeah, that is very important to know about the phenotype. It's influenced heavily by the environment as well. So to kind of further talk about and understand genotype and phenotype, I'll give an example of a taster phenotype. So like in your taste buds, that allows for certain people to taste a chemical compound known as PTC. And PTC, for the people who can taste it, say that it has a bitter taste. And so how this works is you have your chromosome that has the gene for our PTC receptor, okay, which is shown here in the red. So that's going to code for that protein for that taste receptor. And we're going to have different variants of that gene called alleles. And alleles are represented by letters. And the capitalization of those letters, whether it's uppercase or lowercase, matters. So an uppercase letter means that it is dominant. So it means that it is a dominant allele. And a lowercase letter means that it is a recessive allele. So let's assume that the ability to taste, the ability or inability to taste PTC is due to a single gene that codes for that taste receptor. So that's here shown in the red, right? And this gene we get, this chromosome, we get one from our dad and one from our mom, right? Sorry, what happened there? And one from our mom. And basically the combination of our alleles will determine our phenotype. So the combination of the alleles are the genotype, and then our phenotype is going to be expressed by that. So let's say that we have let's represent the taste allele by the letter T. Okay, so, or lowercase t for that matter. represents taste as well. So let's say for an individual to taste PTC, they have to be dominant, meaning that their alleles either have to be a capital T or a capital T and a capital T, or a capital T and a lowercase t, okay? So either one of those combinations will allow for someone to taste taste the PTC. But if someone was not able to taste the PTC, then they would have the recessive alleles. So lowercase t and another lowercase t. So how this works and how you can determine what type of allele is going to be present is through the use of Punnett squares. So Punnett squares are a way to determine the allele combination based on your parents. So let's say that we have our Punnett square right here, and let's say that your dad has a capital T and a lowercase t allele for the PTC receptor, and your mom has also a capital T and a lowercase t. So your dad and your mom are both going to be able to taste the PTC. But if we do this Punnett square here, it is not completely guaranteed that their offspring will be able to taste the PTC. So, like we mentioned before, if we have two uppercase T's, or an uppercase T and a lowercase t as shown here in these three squares, then that means that those are gonna be dominant and the PTC receptor is going to be expressed. But if we have two lowercase t's only represented in one square, then that means that you can't taste it. That means that their offspring won't be able to taste it. And the likelihood of that happening is 25%. And the likelihood of you being able to taste, or the offspring being able to taste it, is going to be much higher at 75%. So this is really important in kind of understanding how the genotype translates into the phenotype, and how the combination of those alleles, or the different variation of those genes contribute to that phenotype.